specifically, I started this company EQO about 2 years ago, and the goal was to bring the modern molecular biology lab to the field of environmental management. Most of my background is in early stage cancer diagnostics and biopharmaceutical development. My partner is a molecular ecologist, and we've got some great business and marketing team people too. So we are a startup here in Austin, and let's get into the science, shall we? So, where are we right now? Essentially, as Stephen and Monica said, right now our best goal, our best chance at anything is prevention. And we'll get into the reasons why. So essentially, whenever these things are introduced into a lake, they usually have to be introduced several times before you get a viable reproducing population. And that's because their method of reproducing is kind of a spray and pray, right? So there's not a great track record of every time something's in. So long story short, you can have them introduced without getting a sustainable population. If you can detect that event, then there's possibilities to prevent them. So what do we do versus kind of, what are we kind of trying to do to introduce, to add more tools for TPWD and for LCRA and other river authorities? This is part of this project we're actually working on with TPWD right now. We were out on the boats all week last week. So essentially what we're doing is, instead of just drawing a plankton net and kind of looking at larvae, we're doing a fractionated series of water. So we do different filter fragments. So we're not just looking for larvae, but we're looking for eggs, for sperm, for if a fish ate it and pooped it out, if there's little cellular detritus floating in the water. We want to know all of that. And not just is it there or is it not, but how much of it is there and what size is it. And from that, we can determine exactly what's going on. Beyond that, what we're working on with TPWD is looking at environmental RNA, not just DNA. So the thing is, if you remember your basic biology stuff, right? DNA to RNA to protein, right? DNA codes for RNA, RNA translates into protein. RNA is not a very stable molecule at all. It breaks down very quickly in the environment. So what we do is we fix it at a certain point and stop it from breaking down. That allows us to take a snapshot to see if anything was alive or dead, if they were metabolizing at the time, if they were responding to treatment a certain way. They use things called heat shock proteins for that. And we can really get an idea of exactly what they're doing, if they're there, if they're alive, if they're dead, if they're early introduction. And that's all for prevention and to aid in any mitigation treatment efforts. So this is kind of the long and short is we have these automated samplers that will be launching in, well, fingers crossed, late July. And the idea is that they contain these filter packs. These filter packs each have a series of filters in them that we can, do what I was just talking about. And they go in, it's fully programmed, so it takes the water sample. So instead of having to send people out on boats every time we want to sample, you just put these in at places of concern. So dams, marinas, that kind of thing. Program it to sample whenever you want, and the marina operator, parks operator, whatever, just pops them out, ships them off to the lab. We do all the analytics and tell them exactly what exactly is going on in their lake. Then it goes back, so that comes out of the box to our lab, where our staff does some fancy magic and wizardry, and we use a combination of right now we're using a technology called qPCR. This is actually a next-gen sequencer in our lab, which we hope to start using for other applications as well, so we can do whole environment analytics, which sounds nerdy, but it's really fun. So let's talk about treatment and mitigation. As Stephen said, there is no silver bullet for this, but we can kind of talk about where there has been success and where we're going next. So right now, most common way of mitigation is physical removal. That's just under an intake, guys. Those are all zebra mussels. And that's with power washing in a boat. That doesn't look like a fun job at all. The other one, this one's real cool, it's called biodiversity exploitation. This is what they've done out in Colorado. So essentially, I'll get into the science of this more, but essentially what happens here is if you catch it early enough and you prevent new species from, new members from being introduced to the society, you can essentially inbreed them to death. So we'll talk about that in a minute. The other one is tarping. This happened at Lake Waco. It seems to have worked. We're getting the data, so we'll find out how well it's worked. In Lake Waco, a few years ago, there was a barge that had come in, and it was just encrusted with zebra mussels and was doing some work around the marina. And as it was being pulled off, it kind of went off the ramp, and they saw that it was encrusted in zebra mussels. So they acted very quickly, tarped off a huge area that basically drops the dissolved oxygen content of where they were introduced. And so far, it's been effective in managing this, and we haven't seen any large populations. Where we are in that project is a question that's better suited to Monica than me, so I'll stop talking about it. There's also chemical solutions. These are primarily only allowed to be used in closed water systems, not open water systems, because obviously you can't be dumping a bunch of chlorine into Lake Travis. And then there's biologicals. These are things like Zequinox, which we'll talk about a little bit. This has fallen out of favor for good reason. And a product that we're working on with U.S. Bureau of Reclamation. So biodiversity exploitation. So first, let's talk about the old adage. This is an updated adage from Dobzhansky. If anybody remembers their 1973 Dobzhansky paper, just me. Okay, the idea was, originally he said nothing in biology makes sense except in light of evolution. So the updated Lynch quote is, nothing in evolution makes sense except in light of population dynamics. And the idea is that if you have a population, you can determine how it's going to change over time based on how diverse the population is and how quickly they breed. So when we talk about biodiversity, I'm just talking about genetic diversity within the population. Whenever a population is introduced to a new area, there's something called the founder effect. You're taking a small subset of the population, introducing it to a new environment, so you are decreasing the amount of diversity in the genetic code, right? And because of this, new populations seldom survive, because they don't have enough genetic diversity to respond to natural selective processes, like say there's a flooding event that changes salinity of the water, or a drought, or lots of other different things. They just can't survive. This worked in Colorado. They had 6 lakes that were infested with quagga mussel, that were noted as infested, and they essentially did a really big push to not allow any additional boats to come into those areas, and they've delisted all of those 6 lakes. They are no longer listed as infested at all. One new one last year was listed as infested, so they're still fighting it, but this is a viable option if you're really careful and really lucky. So we won't talk about these chemical solutions too much. Traditional copper and potash has been used a bit in the upper Midwest to some success, because it's not as environmentally hazardous as traditional copper solutions or chlorine. The problem is with potash, and even with EarthTech QZ, which is a really cool new product that's an activated form of copper, so you can use it at a really low level. You don't have to have a super high concentration. And it kills off the veligers pretty well. So it kills off the larvae, but it doesn't kill the adults. So if you just keep treating it, eventually you can get rid of the adults, because they can't breed. They have a limited life cycle. The problem with this is both of these you can't use in Texas lakes, because we have lots of endangered native mussels, and we don't know how they're going to react to that, and my guess would be not well. So this is something that's being tried in the upper Midwest and out in California and some of the quagga mussel infested sites, and usually lower levels of infestation, but it's not a great solution for Texas. So let's talk about Zequinox real quick. So Zequinox is by a company called Maroon Bio. Thank you. Essentially, it's a soil bacteria that they found produces some kind of protein, they don't know what it is, that happens to kill off zebra and quagga mussels specifically. So their product is they grow up a whole bunch of this bacteria in a vat, kill it off with UV, and that's the product. So one, from a bioengineering perspective, that's not an efficient way of making things. These guys don't like to grow in density, so it's very expensive to get what you need. 2 is, and this is a big problem for me, and why I would, as a guy who used to develop cancer cures, that's not okay. And the reason why that's not okay is you never reach 100%. So all you're doing is selecting for the ones that are immune within the population. So it's incredibly expensive, and then if you go out and treat an area, all you're doing is making it so that the next year you'll have just as many zebra mussels, but they'll be immune to the treatment that you just spent hundreds of thousands of dollars using. Not great news. So what are we doing? This is really cool. I'm really excited about this. Basically, what we're doing is we are working with Bureau of Reclamation, who has developed some antibodies that are specific to zebra and quagga mussels. They were using that for detection. When I heard that, I thought, this is great, because my old company was a company called Molecular Templates. We made biopharmaceuticals for leukemia, lymphoma. And what we did was we reprogrammed bacterial exotoxins and told them, don't kill what you were killing normally, kill cancer. And we did that by taking off their normal targeting domain and engineering on a part of an antibody to reprogram them to attack that thing. We can do the same thing here. And we can do it cheaply. because these are small proteins, and you can grow them up in microalgae, which zebra mussels love to eat. So that's the plan. We're working on it. I have to be clear. This is not coming tomorrow. This is an ongoing in development process. We're at least 3 years away from being able to launch this. So, but it's exciting. So where do we go from here? A lot of additional research needs to happen. We need more support with all of this. We have to have nationally accepted metrics across state lines. Every state manages these things a little bit different. We need to have cohesion as a group. And we have to have a better understanding of how they spread vector to vector and understand how that works. So I just want to leave you guys a little bit of hope. And coming from cancer, you never really hear a cancer researcher say, this problem is, why am I doing this? Throw my hands up, we're never going to solve it. The reason why you never hear someone in that industry say that is because they're incredibly well funded. So they get a lot of wins. You always have a new product that's launching. You always have something that works. This is not an area that has traditionally seen a lot of wins. Hopefully we'll change that and start to have a lot of wins. But right now, everybody involved is doing their best to try and help you guys out. We're all really trying. And hopefully with some additional research and hard work, we'll get this thing solved eventually. But that's the plan. Thank you.
Actually, a couple of these have already been answered because you were talking about lessons we've learned from other places and things that have been tried in other places. Just a quick question. If a bird lands in a water body, can the villagers get under their feathers and be transported to another water body that way?
I get this one a lot, but I think you're better suited.
Yeah, excuse me, I'm going to keep my drink up here so that I got some kind of allergy going on. Birds, they've done a study a long, long time ago. It was one of those old school kind of interesting ones where they had some pools and they kind of herded ducks around from one to another. And they found that the birds, even just moving over the ground, only moved about like 0.1 zebra mussel villagers per bird. And so that's a really, really low risk. There's always a chance they could pick up some vegetation on their feed or move that, but the likelihood of transfer by birds is really low. Boats are really the number one risk.
Okay, thank you. I think one of you said, I think this one did get answered, there are no natural predators here in North America for these things, correct?
Incorrect.
Incorrect, oh,
Incorrect.
Please.
Yeah. That's a really common, thing that people think is that a lot of species get out of control because they don't have any natural predators. They may not have the same predators that they have in their native range, but there are a lot of different species that eat zebra mussels in the U.S. Some of our freshwater drum, catfish, some of the shell crackers, sunfish, carp suckers, carp, grass carp we found them, and some of the turtles will eat them. They just can't make a dent in the population. When you've got a species that grows to 700,000 mussels in a square yard, that's a lot of mussels to eat.
Wow, yeah, thank you. That is a lot of mussels to eat. And they're bottom feeders, people. Don't go out there and eat them. Do not eat zebra mussels. Do we need to say that again? So another question, and I'm curious about this one. So as the population grows in the Highland Lakes, as the population grows in the river, the ecosystem will get changed. We know that and this was actually our recent Barstow Speakers luncheon, we know that there's a lot of interplay ecologically between the health of the river and the health of Matagorda Bay. And that Matagorda Bay, with its oysters and shrimp and so on, has an impact on Austin and the entire state, economically as well. How will these changes in the river's ecosystems impact the bay?
So first, I think the health of the bay is largely dependent on freshwater inflows, water supply. Zebra mussels are filter feeders. They eat plankton, microalgae, as John said, but zebra mussels aren't necessarily taking water out of the river. So as long as we keep having water going down to the bay, and healthy freshwater inflows, I think the health of the bay is going to be okay.
Okay, thanks. Question about what monitoring is happening, what monitoring is happening elsewhere in the state is a big question to answer, but kind of locally, what about Lake Georgetown and Lake Ranger?
So the Lower Colorado River Authority is one of the many partners that's working with Texas Parks and Wildlife Department to monitor zebra mussels, and so all of the Colorado River Basin. Brazos River Authority is working with us to monitor some of these lakes like Granger and Georgetown. And then there are a number of other partners all around the state that are monitoring more than 50 lakes for early detection and for monitoring new infestations. So a lot going on with that. And even some citizen scientist groups in the Colorado River Basin who are helping.
Also, these auto sampling things that we were talking about a second ago, not to push too hard, we are doing a project out at Inks Lake right now, and we'll be expanding that with a prototype that's going in there, and we're hoping to expand those to more areas, and we're also talking with municipalities and whatnot to do additional sampling that way, because there's a lot of municipalities now that are footing the bill for some of these projects. I know Walker Partners, CM Smith, a lot of great engineering firms are on those cases too, and we'll be partnering with them.
Monica, one of the things that you mentioned was that zebra mussel infestations will raise the temperature of the water. Did I get that right? And if so, how does that happen?
In some areas they can, just when they increase the water clarity. And so then, like I said, with the vegetation, increasing the water clarity sounds like a good thing, but it's really not. You have water penetrating deeper, and that allows the nuisance vegetation to grow deeper, and that can be really problematic for recreation and just for lakefront landowners that want to swim near their dock.
And how far downstream will they go? I mean, at some point, salinity, is it salinity that would be the limiting factor going downstream, or is it temperature? How far downstream can we expect to see them appear in the Colorado Basin?
Stephen's already talked a bit about what they've found, but in most cases they found that zebra mussels go about 14 river kilometers downstream. There's a professor at Texas State, Dr. Ashton Schwab.
About 9 miles.
Thank you. Yeah. The conversions for us Americans are difficult, right? So in the river below Lake Belton, Dr. Schwab has found that the mussels will go as far as 64 river kilometers.
40 miles.
All right. So as far as 64 river kilometers, 40 miles downstream, but then when it gets hotter, they die back. So they don't really settle in those downstream areas. They can go downstream long distances when there are big high flow events, but the turbulence and turbidity in the water, the villagers don't have really high survival. So it's definitely a possibility for infestation, but in the rivers they don't usually do as well as in reservoirs.
Okay, thank you. So I didn't hear you say that salinity is that much of a factor.
They can tolerate about 4 parts per thousand. So
Okay, give us a sense of at what point does a Texas river hit that as it approaches the coast?
LCRA would have a better idea of where, but yeah.
It's a lot farther down than 40 miles.
Well, that's why I'm asking. Right? I mean,
Just to hit on the salinity point, they have been found in fairly brackish water in Louisiana. So though we always kind of look for, oh, there's not enough, the salt's too high, or there's not enough calcium, or not enough dissolved oxygen. All that said, it's still somewhat surprising how much they can handle. So, I mean, I don't know exactly where it is on our Texas rivers, but just as a point of reference, they have been found in fairly brackish water in Louisiana.
In some of those cases where, like John said, they've been found in somewhat more brackish water, a lot of that is because there's variation during the day in what the salinity is, and they're not exposed to that salinity all the time.
so like it's the microenvironment where they specifically are within that reservoir or within that water body. So they may find a little pocket of low salinity that they can persist in. Is that what you're saying? Okay, good. All right, what else? Are there additional lessons to be learned from other? I mean, we made you all talk really, really quickly, right? And you probably didn't get to cover half of what you wanted to cover. I mean, are there other lessons from other places that you didn't get a chance to talk about? None.
Well, I would say that a couple of other cases, as far as just referring to the Colorado River Basin that we didn't talk about, is talking about those marinas. And really, marinas need to be gatekeepers to help prevent the spread and protect the lakes, both preventing them from going out and coming in. So the San Jacinto River Authority, for example, has some really good partnerships with their marinas and make sure that everyone has the boats inspected before they come in. And we've talked with LCRA about trying to work on some of those things in this basin. And so hopefully that's something that we'll be able to apply here.
Thank you. Parks and Wildlife has really led the charge in terms of public education with this stuff. I mean, you guys have put a ton of effort into helping boat owners understand what their responsibilities are for preventing the spread. When a water body gets infested like this, how does Parks and Wildlife's role change? Like, is there change in the messaging around the zebra mussels, or is it just kind of doubling down on the, okay, they're here, no really, clean, drain, and dry, it's the law, ... Does that make sense?
Yeah, sure, it makes sense. First off, I'd like to say that Parks and Wildlife may become infested. We work with some of the other agencies, the local groups that are involved, and do a news release. Try to increase awareness, get some signage out on the boat ramps and things like that, and then hand things over to the local river authorities and folks who are going to monitor from that point, because we focus on early detection.
Okay. So one last thing, because people are going to be going out on the water for Father's Day weekend. And actually this came up as KXAN was in here earlier. You know, you've mentioned that if you've got a dock, you got to go in, like physical removal is one of the ways you get rid of it, right? You get in there and scrape them off. When we have a beach or a shoreline area that starts getting a lot of shells on it, is there anything that can be done, or is it just you got to start wearing shoes?
Definitely wear shoes.
Yeah.
And then again, physical removal. I mean, you can tarp those areas, but if it's a shoreline where there's no water anymore, those mussels are going to be dead and they're going to smell. So I don't know if you even want to hang out.
All right. Well, one last round of applause for these folks. Thank you so much for being here.